And then you get to this. This is one of the best stories of all. There is a protein in all of you called p53, and it's a tumor suppressor protein. It prevents the formation of tumors. It's been known for 40 years. But what people have come to realize is that it has multiple functions in cells. And for a long time, they thought that it was just something that it did without any particular reason for it. But now we know that the protein has unfolded parts. Let's go back to here. The N-terminal domain can bind to more than 40 different protein partners because it's a disordered domain of 93 residues. And the C-terminal domain is 100 residues that are disordered to start, and it can bind to an even larger number of targets. We don't even know how many things the C-terminal domain can bind to. And in the middle, there's a DNA binding domain of 200 residues. So the DNA binding domain also interacts with a lot of different targets, and this shows you in the blue ribbons and the red ribbons parts of p53 binding to target proteins all the way around. There's the sequence of the protein, and you can see here's the N-terminal domain binding to a whole bunch of stuff. Here's the DNA binding domain binding to a lot of different targets. Here's the C-terminal domain binding to all kinds of different things. We imagine that p53 binds literally to hundreds of target proteins. One sequence that has hundreds of functions because the N-terminus and the C-terminus are intrinsically unstructured. And that probably saves you from getting tumors even now. Okay, pretty good story. Keith Dunker at Indianapolis, the IUPUI institution, Indiana University, Purdue University at Indianapolis, IUPUI is the name of that school. I don't know how you say that as an acronym, but they don't have a football team. Pardon?
It's Ooey-Pooey.
Ooey-Pooey, that's probably pretty good. So that's where they are, and they helped us with this. They actually gave us this figure so we could put it in our book. But anyway, you get the idea. Crucial transcription factor modulated by a wide spectrum of interactions with lots of other proteins, and it's just incredible. There's a great review about it in 2014 in the Annual Review of Biochemistry, a fabulous review journal. So anyway, tell you one more story as we're wrapping up here, and then we'll be about done. And we'll next time just do quaternary structure for about 5 minutes, and then we're going to be into chapter 7 after the exam, okay? This story is about something called alpha-1 antitrypsin, and it's a protein that's in your lungs right now. And it's a protective protein because it protects you from proteases in your lungs, especially a protease called elastase, okay? And you may not know this about your lungs, but the way you breathe is when you breathe in, you're filling all these little sacs called alveoli in your lungs. You probably know that much. But how much surface area is in those alveoli? If any one of you took your lungs apart and spread all the alveoli, all those little sacs out flat, and put them side by side, any one of you would cover a tennis court with your own alveoli. I'm not making that up, okay? Take all the little sacs, spread them out side to side, and you'd cover a tennis court with your own alveoli. Now, what's crucial is you retain that so you can breathe all the time, right? But this protease called elastase, by nature, degrades those alveoli, kills them, rips them up, tears them into shreds. And if you do that over and over, you end up with chronic obstructive pulmonary disorder, COPD, or emphysema, which is even worse, and you probably don't have a long lifespan at that point. And one of the things that can do that, by the way, is smoking. So I hope none of you do. But smokers oxidize a crucial methionine in the protein called alpha-1 antitrypsin, and that makes things even worse. So let's see what this does. The alpha-1 antitrypsin functions as a molecular mousetrap. It binds the protease called elastase, kind of teases it into saying, come and get the cheese. It's a mousetrap, right? Come and get the cheese, right? Come and take a nibble out of me. And then when you get there, when the elastase gets there, it grabs it and goes whang like this and throws it over to the other side. And we'll show you how that happens. It's pretty cool. It really is a mousetrap. And at the new site, the elastase is inactivated. But if you have defects in your alpha-1 antitrypsin, then the elastase tears up your alveoli and you have degraded lung function. So here's what the protein looks like. The alpha-1 antitrypsin is here, and see it's got a beta sheet in green. See that nice beta sheet? And there's the elastase. And up at the top there, there's a loop of the alpha-1 antitrypsin. I can almost reach it right there. And there's a red beta strand in that loop. And up at the top, there's a methionine. And the elastase needs that methionine to bind to the loop. And when the elastase binds to the loop and tries to cleave it, when it does cleave that loop, the structure grabs the elastase and flips it over to this side, ends up with this structure. And see that red beta strand? It comes along, and that red beta strand fits into the beta sheet like so. So this is the before, when the elastase is nibbling on that loop, and this is after the loop is cleaved, and the elastase is down here, and then this structure is degraded in the cell. So the alpha-1 antitrypsin chews up the elastase before it can destroy your lung. It's there for other functions, but this protein prevents it from doing excessive protease activity and eating your alveoli. So that's the normal function. If your alpha-1 antitrypsin is defective in any way, it polymerizes. And the problem is, it's made in the liver. You may not believe this. Alpha-1 antitrypsin functions in your lungs, but it's made in your liver and is transported to your lungs to function. And if it's defective, if it's mutated, it will just polymerize in your liver and never get to your lungs. And people with that condition end up with COPD and usually die an early death. And one of my favorite biochem colleagues that we're going to talk about in Chapter 9 actually had that condition hereditarily, and he ended up getting a double lung transplant that extended his life by about 15 years. And we'll tell you that story when we get to Chapter 9, so that's a good one. But we'll stop here. And we will, as I say, we'll finish Chapter 9, talk a little about protein folding diseases and structural genomics, and then we'll do quaternary structure. And that's only about 10 minutes next time, and we'll finally be done with Chapter 6. And then we'll do Chapter 7 very quickly, Chapter 8 very quickly, and then we'll do membranes, and pretty soon we'll be doing enzyme kinetics and mechanisms. So thanks very much, everybody. Have a great afternoon. We got office hours this afternoon. Look forward to seeing you there, and come with questions.